this is term 1, week 5, bio-AMS quiz. So let's start. Which of the following statements does not describe the compartmentalization created by the folding of internal membranes within a eukaryotic cell? Basically, guys, what compartmentalization is, so you have your cell, and then each membrane-bound organelle means that each organelle has its own membrane, like its own wall. So each organelle is separated from the other organelles. So inside that organelle, the surface area will obviously increase, okay? And it can have its own pH. It can do whatever it wants inside of the organelle since it has a wall, okay? So like it's not going to affect what's happening in the other parts of the cell. So each part of the cell is its own specific thing. So the one that's wrong is lysosomes in animal cells maintain an alkaline environment compared to the surrounding cytosol. This helps lysosomal enzymes to digest cellular debris. It is not an alkaline environment, it's an acidic environment inside the lysosomes, okay? And you guys should read the other correct ones, just be familiar with them. Number 2. Which structures are common between prokaryotes and eukaryotes? So prokaryotes and eukaryotes, by the way, when you say prokaryotes, we mean no membrane-bound organelles. It doesn't mean all unicellular organisms. The most popular prokaryotes are the bacteria and the archaea. I don't know how to say it properly. This, okay. So the things that we have in common are ribosomes, cytoplasm, and plasma membranes. Okay. The plasma membrane means like the actual cell has a membrane, مش inside the cell. Like the cell obviously will have a wall, but the organelles inside of the cell don't have a wall in the prokaryotes. In the eukaryotes, they have walls. Like they have membranes. Each organelle has its own membrane. Inside of a eukaryote. Inside of a prokaryote, they don't. So inside of a eukaryote, you have membranes for the organelles inside of the cell, and inside of the prokaryotes, there is no organelles have membranes. So unsaturated and saturated fatty acids, basically, when you see a double bond, okay, when you see a double bond, it's for unsaturated. When it's only single, it's saturated. Saturated is solid at room temperature, liquid at room temperature for unsaturated, and unsaturated is 1, 2, 3. So 1, 2, 3, you'll find all of them. This one, you find them in both. You find this in both. So on 1, 2, 3 is for unsaturated, and just 1 and 3 is for saturated. You remember this from the last time. So this is the formation of the peptide bond. You remove the water, you remove the OH, and you remove the H. The H is from the amino group, the OH is from the carboxyl group. You link them together and you'll get a peptide bond. Which of the following accurately describes the molecular structure of cellulose? So cellulose is a polymer of beta-glucose. Guys, you'll have to memorize that. I do not know how to explain it to you. It has beta-glucose inside it, and it's called beta-1,4 glycosidic bonds, okay? So it is beta-glucose. It is a polymer of beta-glucose. Polymer, you know, like polymer is the big chain thing, okay? And the thing that makes up a polymer is a monomer. Okay, and the whole thing is the polymer. Okay, so cellulose is a polymer of beta-glucose. Cellulose is the big thing. The small things are the beta-glucose. The glucose molecules link together by forming beta-1,4 glycosidic bonds. And to form the glycosidic bonds, every other glucose molecule must be flipped over. Basically, so there is no basically, like, you guys have to memorize that. So to form the glycosidic bonds between cellulose, every other glucose molecule has to be flipped over. Okay, so obviously, as we have done many times, NH2 is an amino group. This is the acid group or the carboxyl group, and that bond is the peptide bond. This is a polypeptide, dipeptide. Which statements are correct about viruses? So viruses are infectious agents. Yes, they are. Viruses lack ribosomes. They don't have ribosomes. and viruses are much smaller than bacterial cells. You guys have to memorize that, and you also have to know that viruses can't reproduce on their own. Like they don't have anything to help them live. So they go inside of an organism, okay, and they use the resources of that organism to reproduce and build themselves up. So viral particles can have a DNA or RNA genome that's surrounded by a protein coat called the capsid. So basically, every virus has an RNA or DNA base, let's say, and then around that base there's a wall. It's a protein. It's called the capsid, okay? And each protein molecule making it is called the capsomere. So basically, the small parts of the capsid are called capsomeres. This coat can be surrounded by an outer lipid envelope. So outside of the capsid, you can have a lipid envelope, and it's made of phospholipids. Okay. Prokaryotes and eukaryotes differ in their compartmentalization of cell activities and in their size. Okay. Which of the following are advantages of compartmentalization in eukaryotes? I said that before, but let's go again. Allowing the separation of molecules that could be potentially harmful or toxic from the rest of the cell. So like, for example, in the lysosome, the inside of the lysosome is very acidic. If you didn't have a membrane separating the lysosome from the rest of the cell, it would be very harmful to have an acidic pH inside the rest of the cell. Providing the appropriate conditions for specific reactions. So obviously, if the lysosome wasn't acidic, it wouldn't be able to clean out the cell, because like lysosomes are like little cleaners. Providing more surface area for reactions. So guys, when you compartmentalize a cell, it gives it more surface area to work. It gives it more area to work. Okay. The similar size of prokaryotes compared to eukaryotic cells allows the smaller size of prokaryotes compared to eukaryotic cells. So prokaryotes are smaller than eukaryotes. Allows them to move ions and molecules more quickly from one location to another inside the cell, and to release waste products outside the cell more quickly. As a result, prokaryotes have faster cell metabolism rates and reproduce faster. So basically, the things that happen inside of a prokaryote are much faster than eukaryotes. So prokaryotes reproduce fast and reproduce slow, and do things slower than them. Okay, so we've done this last week. So the pyrimidines are cytosine, uracil, thymine. Those are the pyrimidines. It's one ring. It's a one ring thing. It's a one ring macromolecule, and it has 4 nitrogen atoms, 2 carbon atoms. The purines are 2 ringed, and they have 5 nitrogen atoms and 4 carbon atoms. Okay, and the purines are adenine and guanine, and yeah, that's it. Secondary structure of proteins. So we're just matching. So the secondary structure is the alpha helix or the beta pleated sheet. The tertiary structure is the 3D. It's the 3D shape. It's the thing that ends with the 3D shape. And how does it end in the 3D shape? It's from the hydrophobic interactions between amino acids and water. Okay. Keratin and collagen are for support. You know, collagen for your skin and keratin for your hair. Actin and myosin. Myosin, motion. Motion. Actin, myosin, motion. Hemoglobin and channel proteins. There is a channel, so it transports things. Okay. Identify whether each of the following statements is descriptive of prokaryotes or eukaryotes. So guys, always unicellular is prokaryotic. A eukaryotic cell can be unicellular or multicellular, it depends. Okay. ATS ribosomes, guys, there's ATS and there's 70S. 70S is smaller, ATS is bigger. Obviously, the eukaryotes will have the bigger one, and the prokaryotes will have a smaller one, just because eukaryotes are bigger than prokaryotes. Circular DNA not associated with histones lying free in the cytoplasm. By the way, the DNA of a prokaryote is circular. The DNA of a eukaryote is linear. Okay. Contain membrane-bound organelles. Membrane-bound organelles على طول eukaryote. Cell wall containing peptidoglycan. Peptidoglycan are for prokaryotes, and we have cellulose. No, cellulose. I forgot. Wait, I'll remember. Just give me time. So half-true nucleus or nuclear envelope is eukaryote, And no lysosomes, prokaryote, obviously. No lysosomes, so no membrane-bound organelles, prokaryote. Plasmid is prokaryotes. Plasmid is like the substitute for a chromosome, basically. Viruses are not cells, so viruses are not cells. They have no partially permeable membrane, cytoplasm, or ribosomes for protein synthesis. So, as I said, they don't have anything inside of them that can carry out life. They contain the strand of DNA or RNA, so it depends. They are surrounded by a protein coat called a capsid, and the subunits are the capsomeres, and they range in size. You don't have to really memorize the size. The DNA, which is in the form of a single circular chromosome, is found located in the region called the nucleoid. Okay, nucleoid. Many prokaryotic cells also have extra chromosomal pieces of circular DNA known as a plasmid. So yeah, the plasmid is quite popular. The site of protein synthesis occurs in the ribosomes, so site of protein synthesis always the ribosomes, which contain RNA and protein in two subunits as in eukaryotes, but are much smaller. Okay, so basically the ribosomes in a prokaryote are like smaller. Bacteria are also surrounded by a cell membrane, but their cell wall is composed of peptidoglycan. So yeah, so just like I said, I was correct. So the peptidoglycan, is the cell wall of a bacteria, and the cell wall of a plant, obviously not us humans, the cell wall of a plant is made of cellulose. And even their structure is the same, so it's like they're both beta glucose, beta 1,4 glycosidic bond, blah blah blah. The first cells were prokaryotic cells, so supposedly the first cells were all prokaryotes. A prokaryotic cell gained a nucleus by the plasma membrane, invaginating and surrounding the DNA. Basically, it engulfed it, like the prokaryote ate the, oh my god, ya Allah. A prokaryotic cell gained the nucleus by the plasma membrane, invaginating and surrounding the DNA. So DNA, prokaryotic cell eats DNA, surrounds the DNA, you have eukaryotic cell. As explained by the endosymbiotic theory, the mitochondria evolved when a eukaryotic cell engulfed an aerobic heterotrophic prokaryote which carried out respiratory functions. Basically, what they're trying to say there is the mitochondria formed when a eukaryotic cell, okay, ate an aerobic heterotrophic prokaryote. Basically, the prokaryote didn't have like a, what's it called, it wasn't, it like ate other things. A heterotroph means like it ate other things. Okay, so the eukaryote ate the prokaryote, and now you have a mitochondria. But as explained by the endosymbiotic theory, the chloroplast evolved when a eukaryotic cell engulfed an autotrophic prokaryote. So see here, so it was both eukaryotic engulf prokaryote. Okay, so eukaryote engulfs prokaryote. They both, so both of them, the mitochondria and the chloroplast, both of them was E8P, but the E that ate the P for the mitochondria, the P was heterotrophic. The E that ate the P for the chloroplast, the P was autotrophic. Autotrophic means it makes its own food. Heterotrophic means it eats other things for food. Underline the structures common to bacterium and animal cells. So we both have a cell membrane, we both have ribosomes, and we both have DNA. We did this before. Circular DNA is prokaryote, so circular DNA, linear DNA, I said that. Circular is for prokaryote, linear is for eukaryote. The 70S, the small ones, for the prokaryote. 80S, the big ones, for the eukaryote. Next one, mitochondria and chloroplasts are surrounded by a double membrane. So mitochondria and chloroplast, double membrane, because they got eaten. So one membrane, two membranes. So they already had a membrane, then the thing ate them. The thing that ate them also had a membrane, so now it has two membranes. Mitochondria and chloroplasts contain their own circular DNA. So obviously, the mitochondria, since it was engulfed, it was fine on its own. So it had a circular DNA. It had DNA. And mitochondria and chloroplasts possess their own 70S ribosomes as well. Okay, for the same reason. 70S ribosomes. So bacteria are all unicellular organisms, making them microscopic, obviously. Bacteria are prokaryotic as they do not have a definite nucleus, so obviously they don't have nuclei. Bacteria can also have small loops of DNA called plasmids that float in the cytoplasm. So the loops in DNA that are found inside of prokaryotes are called plasmids. Bacteria have a cell wall made of proteins, sugars, and lipids material, peptidoglycan basically. The cytoplasm of bacteria contains granules of lipids, glycerol, and many ribosomes for protein synthesis. Okay? And please guys, the mitochondria is a membrane-bound organelle, but a ribosome is not a membrane-bound organelle, okay? Please don't get confused. We all have ribosomes, but prokaryotes don't have mitochondria. Eukaryotes, us, we have mitochondria, okay? Also, eukaryotes can be unicellular. Just keep that in mind. So describe the similarities between the structure of the yeast cell and the structure of the bacterial cell. So yeast belongs to the kingdom of fungi. You're gonna have to memorize that. Bacteria belong to the Bacteria belong to the kingdom of prokaryote. I don't know why they spelled it like that. Both organisms are unicellular, so both of them are unicellular. Both cells are filled with cytoplasm and have ribosomes. So everyone has ribosomes. Both type of cells have genetic material in the form of DNA. So they both have DNA. Okay? Now, the differences. No nucleus, bacteria. Loop of DNA, bacteria. Linear DNA, yeast. So yeast is what? Yeast is a eukaryote. Okay? Rough endoplasmic reticulum, yeast. It's a membrane-bound organelle, so it can only be in a eukaryote. No mitochondria, bacteria. No mitochondria, no membrane-bound organelles. Prokaryote is also bacteria. Bacteria is a prokaryote. Plasmids, bacteria. Plasmids are the loop of DNA. Loop of DNA is only found in prokaryotes, and bacteria is a prokaryote. No membrane-bound organelles, bacteria, because it's a prokaryote. Eukaryotic cells have membrane-bound organelles. Prokaryotic cells lack membrane-bound organelles. The nucleus contains the genetic material chromatin, which will condense during cell division into chromosomes. So it starts out as chromatin, then it condenses into chromosomes. The nucleus is surrounded by a double membrane known as the nuclear envelope, which has nuclear pores to permit exchange with the cytoplasm. Okay, so you're gonna have to memorize that. It contains the chromosomes, which carry genes that control cell activity. So obviously the chromosomes are inside the nucleus. The nucleus controls cell division. Okay, so the nucleus is responsible for dividing the cells. Eukaryotic cells, such as plants and animal cells, possess a nucleus. Prokaryotic cells don't. They lack all membrane-bound organelles. So ribosomes are found in eukaryotic cells, including both plant and animal cells, and prokaryotic cells. So why did eukaryote come before prokaryote? Eukaryote came in the sentence before prokaryote because they said plant and animal cells, and plant and animal cells are both eukaryotic cells, and then they put the prokaryote, so everyone has ribosomes again, reflecting the common ancestry of all known life. So we both have ribosomes, which is why they think that we all come from a common ancestor. So this is the 70 and 80S ribosome. So the cytoplasm of prokaryotic cells, 70S. Cytoplasm of a eukaryotic cell, 80S. The mitochondria of eukaryotic cells. Guys, mitochondria and chloroplast always have 70S. Always, always, always. It doesn't matter if they're in eukaryotes. Mitochondria and chloroplast have 70S ribosomes, not 80S, okay? Nucleus of eukaryotic cells, 80S, okay? So everything else in the eukaryotic cell will have 80S. It's just the mitochondria and chloroplast will have 70S. Okay, so guys, these are ribosomes. This is the smooth ER, this is the Golgi body, this is the rough ER. Why is this the rough ER? It has ribosomes on it, the ER with the ribosomes. Oh my god, what the hell? The ER with the ribosomes is the rough ER, the ER without the ribosomes is the smooth ER. The rough ER is for protein transport, the smooth ER is for lipid transport, and that's the lysosome. Okay? The cleaner of the cells, the lysosome. Okay, so this is a prokaryotic cell. Okay, bacteria. That's the chromosome. That is the plasmid DNA. See how it's in a loop. Okay, that's the flagellum. It moves the cell around. The capsule, it's for protection. Cell wall is for protection. Plasma membrane is the thing that lets things in and out. Okay. Okay, let's go through it one by one. So, division by binary fission. Prokaryotes divide by a process called binary fission. Always unicellular is prokaryotes. Okay. Peptidoglycan cell wall is prokaryotes. Okay. Division by meiosis or mitosis is always eukaryotes. Okay. DNA associated with histones is also eukaryotes, and the S motion of flagella is also eukaryotes. Okay. Plant cell have regular shapes. What do they mean by regular shapes? You know how a plant cell has a cell wall and it's like a box? It can't change, but an animal cell doesn't have a cell wall. It's like a plasma membrane, so it can be in like a weird funky shape and like a weird whatever shape, you know. No cell wall for the animal cell, chloroplast for the plant cell, central vacuole, big central vacuole for the plant cell, and a few temporary vacuoles for the animal cell. And the animal cells have centrosomes and lysosomes, plant cells do not. Okay, so match the following structures in the nucleus to the corresponding definition. So the nucleolus is the site of ribosome assembly. So the nucleolus is the thing that assembles ribosomes. The nuclear pore, it connects the nucleus with the cytoplasm, so it's the thing that allows things in and out of the nucleus. And the nucleosome is a semi-solid substance made of water and dissolved substances. It's a liquid. It's a liquid. It's a semi-solid substance inside of the nucleus. Which organelle is continuous with the outer membrane of the nucleus? It is the endoplasmic reticulum. So, like the outer membrane of the nucleus is kind of attached to the endoplasmic reticulum. So, yeah. I did this last week, but I'll go over it again. So this is memorization. So this is the 5' end, this is the 3' end, this is a 3' end, that's a 5' end. That is a bond between C and G. And let me go down. So B is a hydrogen bond between adenine and thymine. So that's A and T. Okay, and that's C, and D is C and G. Okay, and E is a pentose sugar, and H is a phosphate group. So obviously this is the phosphate group, and that is a pentose sugar. Okay. Okay, so tertiary structure is always the thing that results in the final three-dimensional shapes, and globular proteins are an example of a protein with tertiary structure. So the bonds that are responsible for this structure are the ionic bonds between the R groups, the hydrogen bonds between the R groups, covalent bonds between the R groups, and disulfide bonds between the R groups of cysteine amino acids. Only between cysteine amino acids, and disulfide bonds are very strong and stable. By the way, it's like the most stable thing, and it's a type of covalent bond. They asked that last time, so remember. Okay, the primary structure of a protein is the linear sequence of amino acids. So primary structure, linear sequence of amino acids. DNA code for the sequence of amino acids in the protein. So DNA is the thing that holds the code for amino acids. The primary structure influences the final shape and function of the protein. Remember, so the primary structure has a big role in determining what the final shape of the protein will be and function. The bond responsible for the primary structure is the peptide bond. Okay, it's a covalent bond. Bond responsible for primary structure is a peptide bond. Okay, the secondary structure of a protein involves the particular coiling or folding of the peptide chain into two distinct shapes. So the secondary structure, remember, it's either the alpha helix or the beta pleated sheet. Okay, the bond responsible for the secondary structure is the hydrogen bond, so you have to memorize that. Tertiary structure refers to the folding that results final three-dimensional shape, and the quaternary structure is just more than one polypeptide in a chain. Okay. Enzymes are globular proteins comprised of amino acid monomers linked by peptide bonds. You guys have to memorize that. So the enzymes are globular proteins. They are composed of amino acid monomers and they are linked by the peptide bonds. So what is the effective temperature on enzymes? We took this a long time ago. Temperature rises to the optimum, the enzyme activity will increase, okay? Because the enzymes will start moving around and having more collisions. Yeah, just like I said, so at higher temperatures, the enzyme and substrate gain kinetic energy, therefore causing more effective collisions between the enzyme and the substrate. Above the optimum, however, above the optimum, bonds between the R groups of the amino acids are broken. So above the optimum temperature, it's bad, they denature, and after they denature, they can't go back. So denaturation is a permanent change in shape and loss of function of the enzyme at extreme temperature. So, yeah. Oh my god. Guys, I really did this question so many times and I hate it, wallah. So I'm sorry, but I'm gonna skip this one, wallah. I did that one in my last video though, so if you don't understand it, you can watch that one. Tertiary structure refers to the folding that results in the final three-dimensional shape of the polypeptide. I've said that a million times. Proteins that are composed of more than one polypeptide chain have quaternary structure. Also said that a million times. Tertiary and quaternary structure, they're both maintained by the same type of bonds. So it's the hydrophobic interactions, which the hydrophobic interactions is basically the non-polar amino acids, they group together inside of the protein, and the polar and ionic amino acids, they move outside, they position themselves outside, so they're more available to water because they like water, okay? So the non-polar ones, they're hiding away inside of the protein, and the polar and ionic ones, they're not shy, so they're going out and trying to socialize with the water basically. Okay? Ionic bonds are also responsible for maintaining the tertiary and quaternary structure, hydrogen bonds and the covalent bonds, okay? Ester bonds ما دخلهم and glycosidic bonds ما دخلهم. 32. So basically guys, B is the pentose sugar, C is the nitrogenous base, and A is the phosphate group. I cannot help you memorize, I can't give you anything to memorize this, but notice how the pentose sugar is always a pentagon, so pentose, pentagon, you know, and then this circle, and phosphate is always a circle, so I guess that's something, and the nitrogenous base is a square, so yeah. The bond linking the nucleotides in DNA and RNA is the covalent sugar phosphate bonds, okay? It is termed the phosphodiester bond that links the 5-carbon of one sugar and the 3-carbon of the next. So guys, the thing that links the 5-carbon sugar and the 3-carbon sugar is the phosphodiester bond. The bond is formed by condensation, obviously. A water molecule is obviously formed during condensation. It is called condensation. Water is formed during condensation, and condensation builds something up. State whether each of the following is descriptive of DNA, RNA, or both. So DNA polymerase is obviously for DNA. They're both called nucleotides. Each nucleotide contains a pentose sugar phosphate group and a nitrogen containing base. Obviously it's both of them. Consists of 2 strands of nucleotides. 2 strands على طول DNA. Adenine, cytosine, guanine, and thymine is DNA, or like directly. The ribose sugar is for RNA, the deoxyribose sugar is for DNA, obviously. Contains the following, so adenine, cytosine, guanine, uracil is for RNA, then adenine, cytosine, guanine, thymine is for DNA. So uracil and the thymine get switched out. So uracil is for RNA, thymine is for DNA. So it's synthesized during transcription using the enzyme RNA polymerase, RNA. Consists of a single strand of nucleotides. RNA single strand is always RNA, double strand is always for DNA. 5-inch to 3-inch orientation. Both of them have 5' to 3' orientation. Both of them. Whereas the anti-parallel thing, which is one of them is from 5' to 3' and then the other one is from 3' to 5', is DNA. Because like both, there are 2, so 2 is for DNA. Oh my god. Fats are triglycerides composed of glycerol and 3 fatty acids. So you have like basically this, that's what's going on. 3 fatty acids, glycerol. Oils are triglycerides composed of glycerol and 3 fatty acids. So obviously triglycerides are the same in both fats and oils. Phospholipids are the main components of the cell membrane and consist of one molecule of glycerol, 2 fatty acids, and 1 phosphate group. So basically, the cell membrane, not the cell wall, the cell membrane is composed of phospholipids, okay? And that's what a phospholipid looks like. Remember how I said phosphate is always a circle? So see how I drew that circle? That's what it looks like, okay? Steroids include cholesterol, which is an essential component of the cell membrane, and the sex hormones, which include estrogen and testosterone. So basically, steroids, enhance you. They enhance you. Possess only single covalent bonds between the carbon atoms. Single covalent is for saturated. Double covalent is for unsaturated. Okay? Obviously, liquid at room temperature is oils. Solid at room temperature is fats. Obviously, the solids will have a higher melting point. The lower melting point will be for the liquids. The fatty acids are unsaturated, and therefore there are kinks in the chains which prevent the chain from packing together. If the unsaturated fatty acids, okay, it's oils, okay, the saturated ones are fats, okay. So glycerol, A in the diagram above, has 3 hydroxyl groups, okay. Each one undergoes a condensation reaction with a fatty acid, B in the diagram, okay. The final molecule contains 3 fatty acids attached to 1 glycerol by 3 ester bonds. 3 water molecules are released. So basically what they're trying to tell you is you have this glycerol, you have an ester bond. Where is the ester bond? The ester bond. And then you have the fatty acid. So yeah. And then you release 3 water molecules because there were 3 condensation reactions. Guys, I'm sorry, but I did this too many times. Like, I hate this so much. I genuinely can't take this. So again, if you actually don't understand this, then please go back to the video before. But genuinely, I hate this. I hate this topic so much, so I'm gonna skip water. Okay? I did it twice. Periodic video and the MS video. And the last MS video. So 3 videos on. So please, guys. The diagram above represents the structural formula of cellulose. What? Amylopectin. Okay guys, one second. Okay guys, so after I have thought for a bit, I realized that I'm stupid. Okay, so remember how I said that cellulose, each one of them has to be like, every other one has to be flipped over. So look at this. It started on top, then it went on the bottom, then it went on top again. When they change positions like that, when they start flipping the molecules over, it's cellulose, okay? Then down here, notice how these two look very similar, but this looks just like a small part of that, right? Basically, amylose is unbranched, meaning that it's straight, okay? Tamam? And then the amylopectin is branched, so it's bigger. It has like branch, like, understand? And also another thing, this only has alpha 1,4 glycosidic bonds, okay? This only has alpha 1,4 glycosidic. This has alpha 1,4 glycosidic, but it also has the alpha 1,6 glycosidic. The alpha 1,6 glycosidic is the reason why it branches, okay? So just keep that in mind. Water molecules in ice are farther apart than... Okay, this is short. I hate water. Astaghfirullah. I like water. but I hate the topic of water in biology. Water molecules in ice are farther apart than those in liquid water. Farther apart, meaning they are less dense, meaning that they can float on water, meaning that they can be a shield that acts as an insulator for the water under it. Here you have a phospholipid. You can know it's a phospholipid by seeing this phosphate group. It's basically what they're doing is like they just did it like sideways, just how I do it, like this. They just flipped it. So that's the phosphate group, that's the glycerol, and those are the two fatty acid chains. Okay, so see, just like I said, that's the phosphate, that's the glycerol, those are the two fatty acid chains. So, okay, in a phospholipid, the phosphate and the glycerol form the hydrophilic head. So phosphate and glycerol both like water, so it's the hydrophilic head, and then the fatty acids form the hydrophobic tail. You can remember this by basically draw it. Okay, where does what looks like the tail? These look like tails and that looks like a head. Okay, so glycerol phosphate is the head, the fatty acids are the tails. Okay, the tail is always hydrophobic. Okay, it's always running around from things that it so like phobia, tail, runs, motion. So phobia, it's afraid of stuff, so it's always running around, so it's moving. Okay, so tails. And I have no idea what to tell you about the hydrophobic head, so memorize that. So the head is hydrophobic, the tail is oh my god. The head is hydrophilic, the tails are hydrophobic. Guys, I'm so sorry. Guys, the head is hydrophilic. It likes water. It drinks water. Okay, so face, head, drink water. Okay, hydrophilic. We like water. The tails, which like tails, motion, okay, are hydrophobic. Phobia, afraid, you run away from your fears. So motion, tails usually have something to do with motion, I guess. So we'll say that's why they're hydrophobic. Yeah. I hate the concept of water in biology. I'm sorry, but I'm skipping it. Guys, if you want to go back to the water, you can go to I think week 2 AMS quiz. Not the last AMS video. Well, the last AMS video has concepts of water, but the more in-depth one is the AMS video before it. Okay, question 39. Lipids are insoluble in water and include fats, phospholipids, and waxes. Nearly all animals store fats, are more commonly called triglycerides rather than glycogen as long-term energy storage. Fats are also used by animals for insulation. In plants, they are commonly referred to as oils. I cannot explain that. You guys have to memorize that. You guys have to understand that. Okay. Okay, so single sugar molecule, monosaccharide, one single monosaccharide, is a long polymer, polysaccharide, polymer, polysaccharide. Include glucose, fructose, galactose, monosaccharide. Glucose by itself, fructose by itself, galactose by itself, monosaccharide. Maltose and sucrose are disaccharides. They are formed by two things joining together. Disaccharide, di is two. Okay, maltose I think is two glucose molecules, and sucrose is one glucose, one fructose. Please correct me if I'm wrong. Contains two simple sugars that have joined during dehydration reaction. Two disaccharide. Cannot easily pass through the plasma membrane is a polysaccharide. Okay, so polysaccharides are insoluble in water, and the movement of polysaccharides is very hard, so it doesn't easily pass through the plasma membrane. Can be involved in maintaining the structure of cells. That is also the job of a polysaccharide. It maintains structure of the cell. Okay. Why is water important to living organisms? High heat of vaporization, which allows organisms to release excess heat. Again, I don't like this concept, but I'll go over it very quickly since this is short. It is less dense in the solid form, which protects aquatic organisms from freezing during water. You know, it acts as like that little shield. It is a solvent. Guys, Water is the universal solvent. Solvent means water, and what solvents allow us to do is that they allow us to form chemical reactions, perform chemical reactions. Okay, and the high heat of vaporization basically means we sweat. Our body heats up the sweat. Our body heat evaporates the sweat. When sweat evaporates, we feel colder. So obviously DNA, the sugar for DNA is deoxyribose, the sugar for RNA is ribose. That's straightforward. The bases of DNA are adenine, guanine, thymine, cytosine, and then RNA is adenine, guanine, uracil, cytosine. Remember, thymine, uracil switched out. Strands, DNA is double-stranded, RNA is single-stranded, always. DNA polymerase is for DNA, RNA polymerase is for RNA. Very straightforward. Proteins are polymers of amino acids. Each monomer includes NH2 amino group, SOH acid group, or a carboxyl group. What are some characteristics of lipids? Lipids include fats, phospholipids, and waxes. Animals use lipids for insulation and long-term energy storage. Okay, so long-term energy storage, fat, okay. Insulation, basically, the fatter you are, the warmer you'll feel. Fats in animals are commonly called triglycerides. So fats in animals are commonly called triglycerides. If somebody can explain that, like if you can take that sentence and explain it, then you should become a teacher, because I don't know how to explain that. It's a statement, you can't change it, you can't explain it, so there. 45. To synthesize a macromolecule, a cell uses a condensation reaction. So to create a macromolecule, it is a condensation reaction, also called the dehydration reaction. Why is it called the dehydration and condensation reaction? It forms a water molecule. It releases a water molecule. Okay? So that's it. We're done with the quiz. Guys, remember, please study the book and the course revision as well, because there's a lot more questions in the course revision that aren't included in the quiz that might come in the exam as well. Okay? So please remember, always study the book, understand what you're talking about, then go on and do the course revision, and then do the quiz. Okay? Please, guys, genuinely, especially those of you who want to become doctors, biology will be the first thing that your unis look at. So please, focus on biology. Especially for those who want to become doctors, it will be the first thing that universities will look at. Your biology mark. And, well, your chemistry is very important as well. So careful with that. Okay? Thank you so much, guys. Have a nice day. Bye-bye.